Hi, I'm John. I'm the teacher at Beaulieu Convent School, and over the last year or so, we've been investigating daffodils and especially their chloroplasts. So I'll give you a little bit of an overview of our project. If you do have any questions, you can lob them into the Padlet. So in terms of daffodils, they're incredibly diverse. There's some over 20,000 different cultivars, and those are the ones with different shaped petals. Double daffs, we've got ones which have like little orange trumpets or orange cups or white cups. There's loads and loads and loads of different traits that have been selected for through selective breeding over the last probably about 2,500 years. I think the first one was picked out in Greece back around about 400 BC. And nowadays, the I guess the drive, the isn't for diversity, but it's for those Easter and Mother's Day kind of presents and just like house decorations. And that's kind of where the industry about 100 years ago kicked off. Jersey has around about £1 million in terms of an annual turnover for its exports of daffodils. Lots of that goes to the US, most of it goes to England, where it competes with things like Cornwall, Lincolnshire. Um, the UK as a whole accounts for about 90% of the daffodil industry. So daffodils for us in Jersey and for the UK, it's something that's an unnoticed industry probably until you go past a field that's just got hundreds of thousands of daffodils next to them. So what we've been interested in is looking at those daffodils that are on the fringes of fields along roads. So the ones that have escaped from fields, the ones that are not being selected for, potentially the ones that are hybridizing by themselves. And to do that, we've needed to kind of pull in a little bit of citizen science, which is where we get the public, including like staff and students of the school, to go out and take pictures of what daffodils they see. So we've got a little like screen grab of what they've got. This is by no means a complete project in terms of the picture on the right hand side, but it gives you an idea about just how many different types, how many different flowers in particular, how many different you've got the different shapes of the trumpets, you've got the different cup sizes, you've got the different petal ratios. There are loads and loads of things that are just specific to the flower itself. And then you've got the ones which got long stems, short stems. There's so much variation within what is a very, very small gene pool. There's only 56 different kind of historical species. Most of those have been hybridized and lost as specific species through the last kind of 2,000 years, and especially the last 100 years, where we've not kept the heritage of the daffodil, but we've lost it through hybridization, which is kind of quite cool in itself because it allows us to look at the genetics of the chloroplast, which is passed down the maternal side of the daffodil. So our role is to take the chloroplast out of the leaf. So the leaves have a little bit of a cross section under light microscopy. I've kind of brought up the contrast, the light, to really show you the prevalence of the chloroplasts in these leaves. We then, working through centrifugation this year, is an extra little step. We're isolating the chloroplast and we'll then do a protein digest before sequencing it using nanopore technology. Last spring, we didn't do that step. We were using a bigger, what's called a flow cell, which takes the strands of DNA. I'll show you a video of it a little bit later to sequence. When we didn't, we were getting around about 800,000 reads from our sample from 0.1 grams of leaf tissue. But in there, we also had things like the fungal, everything that was airborne that landed on the leaf kind of got on there. Any kind of diseases that were embedded within the leaf. It's again, we had to filter out the reads, and to do that, we do what's called a reference alignment. where we take our rates and compare it to the only other chloroplast, well, daffodil chloroplast genome that has been sequenced, that was only published in 2019. So I'll just show you how the nanopore works. So this thing is about the size of a Mars bar, and I have put a flow cell on it. That flow cell has a tiny little hole at which we can pipette our DNA, and it's on here. These little pink bits are pink, and the white bit are the adapter, and a little motor which ratchets the DNA as it comes into this flow cell. Now the DNA is double-stranded, which isn't particularly useful. So whenever it hits the pore, it gets ratcheted into single-stranded DNA, and it gets passed through one nucleotide base at a time. And it's the difference in charge that across this membrane that is interpreted by the sequencer itself, which is an incredible technology. The subtleties of the differences. And then we take our little bit, and we have thousands and thousands and thousands, and we create what's called a consensus, what we're confident in terms of the sequence. You can see like there's lots of bits that maybe didn't align very well, maybe they're not aligned, maybe they're not even chloroplast DNA. And this is why we need lots and lots and lots of repeats. The longer the reads, potentially the better, but nanopore technology is around about 95% confidence level for the technology. So our aim going forward is to try and get to something like what we have on the right hand side, where we've got a single genome correctly assembled and correctly annotated. And if we don't get there, it's not the end of the world. That's the dream. But we'd also like to be able to get enough regions of a chloroplast that is comparable between different types to then start looking at the phylogeny, looking at how closely they are related to each other, and maybe trace back as to their heritage as well. So by all means, if you do have any questions, do let me know through the Padlet, and I'll do my best to answer them.